We're next going to hear from Deb Fuller, who's professor in the Department of Microbiology at the University of Washington School of Medicine and Associate Director of Research at the Washington National Primate Research Center.
That's right. Great. Thank you for inviting me to talk today about our work to look at the feasibility of developing a nucleic acid vaccine for valley fever and also a non-human primate model. I have a couple of disclosures. I'm going to talk about two platform technologies, one of which a company I'm a co-founder of, and the other one in which I serve as a scientific advisor. So I'm a vaccinologist, and I've developed vaccines for a lot of different infections, mostly viral infections, but also bacteria and malaria. And they all hold in common the same things in terms of what we need to develop an effective vaccine. One is, first you need to know what types of immune responses are going to provide protection, and then you need to know what parts of the pathogen or antigens these immune responses need to target. Then you need a vaccine strategy that can induce those responses. And finally, you need an animal model that closely models human disease to test that vaccine in. What I'd like to actually talk to you about today is how we can use nucleic acid vaccines, potentially maybe the right kind of vaccine strategy for valley fever, and that non-human primates could play a critical role in helping to develop the vaccine. And by developing these two tools, we may be able to get around to addressing these two questions here. So first about nucleic acid vaccines. Now, as you all know, the mRNA vaccines have emerged to the forefront of our knowledge through the COVID-19 pandemic. But myself, along with many other investigators, got started on nucleic acid vaccines over 30 years ago. And their success for COVID-19 was no surprise to me or to my colleagues. Even 30 years ago, we knew that they would be really promising approaches to quickly address emerging infectious diseases and pandemics. I like to say that developing an effective mRNA vaccine was a turning point for COVID-19, but COVID-19 was also a turning point for nucleic acid vaccines, meaning it's put them really on the map. In many respects, I think COVID-19 was a low-hanging fruit for nucleic acid vaccines. If you think about those 4 points for SARS-CoV-2, we knew exactly what to target. We knew exactly what kind of immune response it was needed. We knew nucleic acid vaccines could induce those responses, and we had an animal model to test that in. And so I just kind of want to cover some key advantages of DNA and RNA vaccines and why I think they might be really particularly effective for a valley fever vaccine. One of the first things is that they really only need the genetic sequence of immunogen to make a DNA or RNA vaccine. We can design an RNA or DNA vaccine and have it in animals within 7 days. And so that actually gives a nice advantage in terms of rapid design and development. But it also allows us to precisely design the vaccine so it focuses immune responses only on those antigens you need, and conversely avoids those that might enhance disease or cause high reactogenicity. It enables rapid discovery of new immunogens, and I'll get to that in a minute. But it's also highly amenable for delivering multiple antigens together. And it has a very high safety and low reactogenicity profile. DNA and RNA vaccines, from the first experiments we did back in the early 1990s, it became very apparent they were very effective in the induction of Th1 type immune responses as well as CD8 positive T cell responses. That's sort of their hallmark. And it's something that in many respects earned them the reputation of being very much like live attenuated vaccines, but without the risks associated with them. And if that's not enough, we can actually co-deliver genetic adjuvants to modulate and skew the response in whatever direction we want. Finally, I'll talk about a little bit about how they can be used to induce a mucosal immune response. So nucleic acid vaccines, as I mentioned, are very effective for inducing the types of responses likely needed for protection from valley fever. And these responses, the Th1 and Th17 responses that Dr. Hung just talked about and that she wrote about in this review, are precisely the responses that mRNA and DNA vaccines are very effective in inducing, in addition to CD8 positive T cell responses, or CTL, that could also play a role. Now, nucleic acid vaccines also enabled rapid discovery of protective immunogens. If you think about traditionally how we actually do sort of discover new vaccines, we might take known immunogens or virulence factors or epitopes, and we encode them in plasmids, and then we'll produce the proteins and inject those into the mice. But often the bottleneck is the production of these proteins themselves. And the way nucleic acid vaccines can actually bypass that and you can just directly inject them into the mouse, thereby shortcutting that process. And as I mentioned, it only takes me about 7 days to make a library of plasmids to inject into mice. There are differences between DNA and RNA vaccines I want to get into because I'm going to talk about both of them. And the first is that immunogenicity of RNA vaccines has proven to be better. And that makes sense from the perspective that DNA has to be delivered in the nucleus, whereas RNA only has to get into the cytoplasm. So you're going to get a lot more cells expressing RNA and a lot more protein and a lot more immune response. RNA also has self-adjuvanting properties. It's a natural type 1 gamma interferon inducer, whereas DNA vaccines tend to be poorly immunogenic unless you co-deliver them with a genetic adjuvant. On the other hand, DNA vaccine stability is much, much better than RNA. And I say if we can make DNA work as well as RNA, then they would actually take over for RNA. DNA vaccines are stable for years. You can put them on the shelf, whereas RNA vaccines are unstable. As we learned, the mRNA vaccines required ultra-cold refrigeration to make them stable. Finally, in our lab, we actually are investigating them in parallel. And what we found is that DNA and RNA vaccines can induce distinct types of immune responses. This shouldn't be a surprise because cells sense DNA and RNA differently. It depends on the antigen, but in some cases, we'll find that RNA tends to be better at inducing an antibody, whereas DNA tends to be a little bit better at inducing T-cell responses. And so I'm not going to get into this today, but we are working in our lab and trying to leverage them both, potentially in combination, to induce a broad array of immune responses. So let me just talk a little bit about our nucleic acid vaccine platforms. The first I'm going to talk about is a self-amplifying RNA vaccine delivered by a novel lipid nanocarrier called Lion. So you're all familiar with mRNA vaccines. Replicon RNA or self-amplifying RNA vaccines are similar in the sense that you deliver RNA, but they include an additional code for this protein called replicase that instructs that RNA to make additional copies of itself so that you actually have more RNA copies. More RNA copies makes more vaccine antigen. In addition, this process of replication induction is actually a self-adjuvanting property. So you get away with a lot lower doses of self-amplifying RNA will induce substantially greater responses in the mRNA vaccine. So the other platform that I'm going to talk about is a gene gun delivered DNA and RNA vaccines. This is a needle-free technology that delivers DNA and RNA directly into cells of the epidermis. And the big advantage here is that it targets the epidermis, not the dermis. It's not an intradermal delivery, it's epidermis. And why is that important? Because the skin is a very immunocompetent organ, and most of the antigen-presenting cells are located in the epidermis. So it's a great inductive site for immune responses, and this combined with the direct intracellular delivery of the DNA and RNA makes the gene gun a very efficient approach. So let me first talk about the Lion formulation with our self-amplifying RNA vaccine and why it's different from the lipid nanoparticle type of formulation that we have with our COVID-19 vaccines. Here's a lipid nanoparticle formulation, and so your Moderna and Pfizer vaccines are going to encapsulate the mRNA inside the lipid nanoparticle. Now, this encapsulation process is not trivial. It's actually a quite complicated process to successfully do that. Once it's done, it is not very stable by itself. It has to be frozen at ultra-cold temperatures or it would degrade pretty quickly. This formulation then is also what we call married to the RNA, meaning you have to formulate it, and then if you want to change it out, you have to go through the entire formulation again. Likewise, in addition, lipid nanoparticles delivered with mRNA vaccines actually result in systemic distribution. They get injected in the muscle, but they do distribute all over the body, and sometimes that can have some safety concerns, and that's in part why they may have a little bit of reactogenicity. So we, in collaboration with HET Bio, developed a new formulation. It's called Lion and differs from the lipid nanoparticle in that the RNA is not encapsulated. It sits on the surface of the nanoparticle. What that enables is us to stockpile this nanoparticle formulation, and then you just mix the RNA with the lipid nanoparticle one-to-one right before you end up using it. And so that allows this flexibility, this plug-and-play functionality, so that if you have to update the RNA or change it out, you just pull out a sock of the lipid nanoparticle and you mix it on site. So this formulation is also very stable, much more stable than the lipid nanoparticles at 5 degrees Celsius for over a year. And notably, it can be lyophilized, and so then that actually increases its stability, and then within 5 minutes be ready to inject. And then this liquid formulation is still stable for 3 weeks at room temperature. Importantly, there's local retention at the injection site, which means it doesn't distribute all over the body, and that might have enhanced safety profile. So I want to emphasize again that replication or self-amplifying RNA is distinct from mRNA. We already covered that it expresses more protein, it has self-adjuvanting properties, but an important aspect of it is that it induces stronger immune responses with lower doses, down to 5 microgram, and lower reactogenicity at high doses, at 100 micrograms. Now, your Moderna and Pfizer vaccines, their optimal dosing is between 30 and 100 micrograms. 100 micrograms is a limit. You can't go over that, otherwise you have too high of a reactogenicity. And so if you've got your bivalent vaccine, you've got 50 micrograms of the new variant and 50 micrograms of the other one. If they have to add in another one, now you got 30 micrograms, and you're at the lower limit threshold of where those antigens are going to be immunogenic. In contrast, this particular technology allows us to co-deliver multiple different RNAs because we can get away with much, much lower doses. And so as a proof of concept, we actually tested this replicon RNA vaccine as a pentavalent vaccine co-delivering 5 different antigens for respiratory infections. And what you can see is that each of these different vaccines are inducing comparable immune responses to when they're alone or in the context of the pentavalent vaccine. And so this is important, we think, because we think an effective valley fever vaccine is likely going to have to be multivalent to target the pathogen at different stages in its life cycle. So let's talk a little about the gene gun delivery. It's distinct from electroporation. Electroporation is another method for delivering the DNA. And gene gun is pain-free. It results in direct intracellular delivery in the highly immunocompetent skin. And importantly, when you compare that directly dose per dose electroporation versus gene gun, you get much higher immune responses with the gene gun and better protection. One of the other aspects, of course, is that it can deliver multiple DNA vaccine constructs in a single dose. We do this by actually formulating the different constructs in separate gold beads and then mixing them and delivering them together. So the gene gun can deliver both DNA and RNA, and it should look at no surprises at in vitro delivery, where you see a lot more cells expressing RNA than the DNA, and that results actually in gene gun delivery of RNA inducing better immune responses than gene gun delivery of the DNA. Importantly, the gene gun also can induce mucosal responses. In this study, we're comparing electroporation delivery of 1 microgram to gene gun delivery of 1 nanogram of DNA. We get comparable immune responses in the systemic, but we get much higher mucosal responses in the lung with the gene gun. Why is that? We don't quite understand, but it's a well-known phenomenon that the skin can serve as a robust inductive site for mucosal immunity. And this actually played out in a study where we actually investigated an influenza DNA vaccine designed to induce T-cell responses and induce robust mucosal T-cell responses in the lung. It blunted the viremia and accelerated clearance, and we ended up showing that that protection correlated with the frequency of mucosal T-cell responses in the lung. So we think this could be important for valley fever in terms of providing protection at the site of exposure. And so finally, we're actually comparing these 3 platforms, gene gun delivered RNA and DNA, to IM delivery of replicon RNA in lion. And what we actually see is the IM delivery of the replicon RNA in lion induces responses quicker, but after a booster immunization, all 3 of these technologies are inducing comparable immune responses in non-human primates. Finally, I just want to emphasize that these technologies have been into phase 1 human trials. The replicon RNA vaccine was recently licensed for human use in India, becoming the first self-amplifying RNA vaccine for human use, and the gene gun delivered DNA vaccines have been in multiple human clinical trials. As one final point, I know I'm out of time, I just want to talk about the development of the non-human primate model for valley fever. So non-human primates in the Mesa site are being bred. They are pigtail macaques at the Washington National Primate Research Center, and these animals get naturally exposed to valley fever, and when they do, they exhibit pretty much precisely the same symptoms that humans do, and show diverse clinical presentation. Then the affected animals can be effectively treated with antifungals. And so this actually provides an opportunity for us to think about using non-human primates as a model for valley fever. They actually do respond to a lot of other treatments. Here's an example of using flucosol impregnated beads to eliminate bone-infused cocci infection and actually clearing it from that animal. What's important is that we are developing an experimental pigtail macaque model in collaboration with Bridget Barker at Northern Arizona. And what we're hoping this would do is provide a highly relevant preclinical animal model to test new vaccines and therapeutics. And I was looking, a lot of the different sort of things that were talked about here, they could be used, the colony, we have a lot of specimens from them to be used for diagnostics, for other sorts of applications. These animals are constantly tested every 6 months, and when they come up positive for antibody, regardless of whether they have symptoms, they are treated with the antifungal. So this is actually an example of a colony getting treated, even before symptoms occur. It might be interesting for study. And if you're interested in actually getting samples from these animals, I'd be happy to put you in contact with people at the Washington National Primate Research Center. Finally, we really want to address these important questions. What are the earliest immune responses to infection? We can do that in an experimental model for non-human primates, and what are the mechanisms driving these different outcomes in these animals. And thank you.
Thank you very much.